Hello, my name's Adam Rutherford, and we're here at the Kavli Prizes 2016, and I have the privilege of interviewing the Neuroscience Laureates for 2016. Eve Marder, Carla Schatz, and Michael Merzenich have spent their lives and careers unpicking how the brain changes as we develop in the womb and also in adulthood, yet at the same time, how it maintains itself in a reliable state. And in doing so, they have helped us redraw our understanding of how the brain develops, but how it treads that delicate balance between maintaining itself in a stable state. Now, I come from a generation of scientists for whom brain plasticity, the idea that brains can change in adulthood, was just accepted and normal. But all of your work, all three of your careers, have effectively been changing that idea for people like me to accept that. Is that right? How much of a challenge was it to understand that the brain was plastic?
We're looking at Mike.
Right. Well,
he had the challenge to start with in terms of the adult brain.
Right. Well, it was the dominant. There was a group of scientists from physiological psychology that were arguing on the basis of studying classical conditioning or Pavlovian conditioning that the brain did have a capacity to change in adulthood, but in the mainstream of science, this was not accepted into the 1980s. And then we and others did experiments in the 1980s that demonstrated that large-scale plasticity could and did occur. And ultimately that demonstrated quite quickly that any time you acquire a skill or an ability, that acquisition is really a product of the remodeling of your brain that occurs as a consequence of brain use. We also begin to understand that what differed in an older brain as opposed to a younger brain was that plasticity was closely regulated by processes in the brain that only allowed change when, in a sense, it was to the purposes of the brain. That is to say, plasticity was controlled as a function of the state that you were in when you were engaged in an activity or a behavior, and as a function of outcome. So in a sense, you move from a young period where the brain has more rampant capacity to change to an older age in which change is still occurring on a large scale under the right conditions, but only under the right conditions, and change enabled as a function of outcome.
It seems so obvious now, though, that the brain would be something that responds to the environment, and that's how we form thoughts and memories.
But I think what happened too is that obviously we can't learn and remember unless the brain has the capacity for plasticity. And I hope that we're all still learning and remembering. And I actually think it's fun to think about the idea that after you hear us talk, your brain will have changed as well.
Right.
But the idea that the brain was not plastic in adulthood, I think also was affected by the discoveries, again in the 70s and 80s, that there are these early developmental critical periods where learning, as Michael said, is rampant, is happening very, very rapidly. And then it's very clear that those periods trail off. And some, I think, misinterpreted the science behind the discovery of these developmental critical periods to think that these were periods and then the window slams shut on those periods, leaving us all without the capacity to have any brain plasticity. And that really did influence, I think, thinking about the adult brain for a long time.
And now we think about those times of, as you say, rampant development, and there's growth of new neurons, there's new synapses, and there's also pruning happening at that time. But back when you were first exploring this area, and I suppose this question goes to all three of you, was there resistance to the idea that this kind of plasticity was not abnormal in later stages of life, after childhood development?
I mean, I think it's important in talking about plasticity to understand that there are many different mechanisms of plasticity, and plasticity is important in the ordinary operation of neural circuits as well as developmental and major modifications of brain structures. So I think that what happened starting in the 70s is that people started realizing that there were plasticity mechanisms that were operating on all different kinds of time scales, such as the neuromodulatory time scales that are important for changing the state of circuits so that they can react differently under different kinds of environmental circumstances. And then that segued into, at some point, understanding that many of the same mechanisms that were called into play in what we would classically call learning were some of the same mechanisms that were classically thought to be important in development. And these guys were really important in that. I think that, however, it's really important to remember that for different people, plasticity covers a myriad of different kinds of mechanisms.
Sure. You talk about mechanisms a lot, and we do in developmental biology, but that, I think, is a crucial concept in the sense that it's neurons making contacts, isn't it? It is actual, the migration of axons through tissue that is changing, at least for some of these.
Well, I was going to say, maybe we ought to define what we mean by plasticity.
Please do.
We also might have different definitions, but mine is a very simple one, which is how brain circuits change with use and with learning. And use can be generalized also, because there are different ways that circuits get used, and use can actually start even in the womb, in the embryo, which is rather surprising. This is the other unexpected, I think, discovery, which is that brain circuits are plastic even before birth, and they're being used before birth.
And that's in your work. You've focused on the retina, and obviously that embryos are not using their eyes to see things, and yet you're seeing this effect, this connection between the cells in the retina and the visual cortex.
Yeah, so that's why I think my definition is very simple, because the eyes are sending signals to the brain, and those signals are being used to remodel circuits. And the circuit remodeling is brain plasticity during a very early period of development, when using that circuit is required to get the adult pattern of connections to form. So it's all about, the functional and structural remodeling of the circuits. So I don't know what everybody else means by plasticity.
Well, it's very interesting, because if I take the words that Carla just used, those words could be used to talk about the more temporary or reversible kinds of plastic mechanisms that take place as you look at a brain during the day and then a brain at night, where you have cyclical hormonal changes and neuromodulatory changes that actually change the state of circuits, but not necessarily permanently, but as they function in behavior. So I think all those same words that Carla just used could be also employed for shorter term plastic mechanisms, which are very, very important for the way circuits are built so that they can perform in behavior.
And in your work, you've been looking at things like natural cycles, and as you were saying, so those plastic mechanisms are occurring during just sort of standard functioning during a day, rather than forming new memories.
Some of those, and I would just like to say the other important concept that I think is important to introduce, and I think we would all agree about this, is that the neurons in the human brain in a really healthy person may live for 100 years, but all of the protein molecules that are important for cell signaling are turning over very, very rapidly. So in all of the changes that are taking place that allow you to incorporate learning and experience into brain structure, you also have to remember that the brain must be constantly rebuilding itself. And so it's constantly rebuilding itself to maintain the stability that we want in a normal, healthy adult brain, but using molecules that are constantly being replaced. And so that's what's really interesting, is this enormous numbers of different time scales that have to be called into play to do all the different kinds of things that we've been talking about.
And these models of plasticity that we're talking about, are they? Do you agree with these sorts of definitions in your work, Michael?
No.
Oh, good.
No, what I believe is that there's much more than circuit refinement that's occurring. There are a whole long list, a sort of laundry list of chemical and physical changes that are occurring as the brain changes itself, as it revises its operational capacities. So a whole series of things are occurring in the background. And so the brain is, the way that brain fibers are myelinated, the way that trophic factors are controlling changes in brain processes, the way that whole series of things are changing in parallel with changes in effective circuitry. And really, the plastic
But that
consequences.
is all effective
Yeah, I
circuitry.
think we're
Yeah.
talking
I
about
don't-
the same thing.
Right.
But what I'm getting is that the dynamic process of
Right.
how the brain is
Yeah.
constantly changing
And I'd like to add,
is shared.
I'd also like to add something to the question you asked a minute ago, and that is, was there resistance to this notion? And when we first published plasticity experiments, we first submitted them for publication. We got colorful responses back from reviewers that said this can't possibly be true, that we can't see how we can deny this, but can't possibly. This occurred several times. We had strong resistance when we reported this at scientific meetings. People stood up in the audience and said, this just can't be so. This is not so. So there was very strong resistance. And partly it was because the experiments that seemed to argue that the brain basically came to a sort of hardwired state early in life were so compelling. They were so beautiful, so elegant, so believable, so convincing. Even I was convinced for a long period of time until you could say we stumbled under the fact that the brain, in fact, had this rather remarkable capacity to change at an older age under the right conditions. And that was one of the critical things that how the science evolved. We began to appreciate the enabling conditions that were required for older age plasticity to be in play. And really people imagined the marvelous plasticity that occurs in an infant as being critical to having a well-organized life. But in a sense, the brain evolves so that plasticity at an older age is purposeful to the brain. It's now in a wonderful state. It's under the brain's own control. And what really happens across early life is the brain evolves the capacity to control its own change, which is sort of a wonderful and miraculous thing.
Unless the brain changes as a result of insult or injury.
Oh yeah,
in which case,
yeah, bad things do happen in brains and life.
And in terms of understanding the plasticity and the way that brains change in response to injury,
Right.
has that informed a lot of your work?
Oh, absolutely. No, that was a critical aspect of what, and of course this is a thing of great interest because there were a parallel understanding of the phenomenology of this that had been derived from the psychological sciences and from rehabilitative science and from neurology. So this was a great contributor to our understanding of plasticity processes, especially early in their study in adult brains.
It's kind of interesting that on top of the kind of plasticity that's possible to tap into through training, and as shown in much of your elegant work, on top of that, there appear to be breaks on plasticity because what my lab, but other labs have found, is that it's possible to take those breaks off in the adult brain and restore more juvenile-like, exuberant plasticity to the adult. Now, we don't really know if that's a good thing or bad thing, because this has been mainly done in animal studies. But the idea that there might be even more plasticity in the adult brain that we could tap into and manipulate, particularly for recovery and for treatment and for retraining, is kind of exciting.
So introducing a sort of a more embryonic state.
Yeah. So it's wonderful because the development has helped to inform the adult condition.
But I think it's important to, sorry to interrupt, is to understand that the brain has multiple ways it can do almost anything important. So it's not like there's a single pathway from X to Y to Z to ABC. The brain has often many parallel pathways by which two groups of neurons can interact or by which the cortex can influence the spinal cord. So sometimes after lesions, what you can do is sort of reroute. So you may have, one of four pathways may be gone, and usually that might be the dominant one, but maybe you can learn to use alternate pathways to actually do what looks like very much the same sort of pattern of behavior. And I think that's what's really important, is learning how the brain can control which multiple pathways it uses under which circumstances.
What I'm getting from all three of you is that the brain is much cleverer than we thought it was.
The brain of a crab is much cleverer than I am. Well, so,
why don't you tell us about that? Because a lot of your work has been on crustaceans and crabs and lobsters, and that's not obvious to many people why they would be of great interest to a neuroscientist.
Well, there are advantages for working with what used to be called simple systems. We've learned that none of them are simple, they're just small. But sometimes when you work with a small system like the one I work with, or that people work with C. elegans or flies, when you study the same group of neurons over and over and over and over again, eventually you start seeing general principles that are true, presumably, in mouse and cat and human brains as well. But those principles can be seen more clearly because those circuits are better defined and the neurons are well identified and they're large enough to manipulate. And so the reason why we've been able to study some very fundamental and large principles was because we were studying a very simple circuit.
I don't want to reduce this to the prosaic, but I've spent my life inside molecular biology labs, and I've never seen a lobster in a lab, so I can't quite picture what the actual physical setup is like.
Well, down in the basement of our building, we have 12 or 15 little seawater aquaria. The animals come out of the Boston Harbor. They actually take a taxi to our lab.
So they're local.
They're local, and then they go into the tanks, and then they come in an ice bucket up to the lab, and we carefully dissect them, and we put the edible parts of lobsters in the freezer, and they do get eaten, and the rest of it.
I'm not sure that's legal. But the point is, as you're saying, that it's not that you're interested in the lobsters per se,
No.
it's that they provide a way of answering fundamental questions about neuroscience.
Yes. No, I mean, I think what's really remarkable about these animals is that they have neurons and circuits that are really, really beautiful and amenable for understanding.
And well characterized. Now, Carla, there's a phrase that you've been associated with that we use in neuroscience and genetics all the time, which is that cells that fire together wire together. And it's a great little maxim, but perhaps you could explain what it means and why it's such a significant idea.
Yeah, and the other part of that is out of sync, lose your link.
Okay, do the first one first.
Okay, so the first one, cells that fire together, wire together. So the idea is that if two neighboring cells are active at the same time, let's imagine they're placing telephone calls to, to New York City or to Oslo, and those phone calls are received at the exact same time by the target cell. The connections actually strengthen, so you get a much more reliable line from let's say the eye to the brain or New York to Oslo. On the other hand, if cells are out of sync, so that a phone rings first from one cell and then from the other cell in Oslo, one of those connections may strengthen, but the other may be eliminated. And these funny little sayings actually represent knowledge that we have about the cellular mechanisms of how connections, the synapses between neurons, how those connections are strengthened or weakened with use. And it's telling you that the pattern of use, the way that the information is coming into the target cell, is critical as to whether or not the connections are going to be strengthened or weakened.
And that occurs in your work as a developmental biologist. That is something that occurs pre-birth in the retina.
Yeah, I mean, that's very surprising. I think that the thing that's rather surprising, and by the way, that's what we would call a cellular mechanism of plasticity. So then we're actually, sort of diving down into how plasticity works and how the strength of connections change. But what was rather surprising is to find that this cells that fire together, wire together mechanism work early in development, even before birth. I think people thought this was a mechanism that worked for adult learning and memory, where it was first discovered, But now these mechanisms are much more broadly understood to happen throughout life.
It makes me wonder how that system might have evolved, that something is becoming functionally significant even though it's not
Well,
having a function at that point.
I mean, I think you have to remember that when a child is born, they have to breathe as soon as they're born. And you can't wait for experience to build that respiratory circuit. You have to build it so that it works exactly when it's needed or considerably before you think it's needed. So I think, everything that's really important, you have to imagine building, or at least building the scaffolds of, before the animal is born.
Now, the Kavli Prizes are not for applied research. They celebrate basic research, pure science, curiosity-driven science. But it's quite hard to have this conversation without immediately thinking of potential clinical relevance for the discoveries that you made. And let's start with you, Carla, that what might be, down the line, the clinical relevance of understanding things like how neurons in the retina wire up with the brain?
Yeah, the surprising thing really for us is that there seem to be many clinical connections, and it took many years of trying to study how the eye and the brain connect to understand the relevance. But if you imagine that all the time there are decisions during development being made about which connections should be strengthened and which ones should be eliminated, and this is part of a learning process that even starts before birth, wouldn't it be great if you could discover molecules that are controlling how the connections get removed or how they get strengthened? And for us, we were very surprised to find that we could discover molecules that were important for removing connections during development, the ones that weren't necessary, the ones that weren't used in the kind of firewire, or I like to call it, use it, lose it, process. Those get eliminated. And sadly, in Alzheimer's disease, for example, many connections get eliminated. And some of the molecules we've discovered seem to be implicated as culprits also in this elimination process that happens in Alzheimer's disease. So there is a very tantalizing connection, for example, there. Another connection would be in brain retraining with recovery, recovery of function, because again, use it or lose it, and somehow the brain plasticity that we study in development may come into play again during recovery and training after things like stroke. But this is where Mike's work really becomes highly relevant.
Well, sure, I want to ask that question to you, Mike. That I think yours is possibly the most obvious connection between clinical relevance and basic neuroscience.
Well, we spent quite a few years in my research group, and all of the science that we do, of course, is team science, and trying to define the conditions under which we could control brain plasticity with high efficiency and high effectiveness. And then we've tried to translate that out to help people that struggle, primarily to help people that struggle. And we've applied it now in about 7 or 8 million children and adults that, for various reasons, are struggling in life. And these are children that start out in life with struggling to learn, to develop their capabilities in a normal way, or adults that are either slowly regressing in life on the path to dementia or Alzheimer's disease, or individuals that have had suffered from psychiatric or neurological conditions that impair or degrade their lives. In every one of these instances, it's been shown over and over and over again, we can drive changes in the brain by targeted training that both restores their capabilities as we would define them on some level, functionally or behaviorally, but also we can drive substantial changes in their brain health. So if you think of the organic health of the brain, like you think of the organic health of any other organ of the body, we can drive changes that clearly translate to a recovery of health. And what this means to us is that brain health can be managed, like you can think of cardiovascular health being managed by engaging the brain, you could say, to sustain itself, to sustain its assets, to sustain its capabilities, its functionality. So we know now that we can substantially delay the onset of dementia, substantially decrease the probability that dementia will arise in an older life. We now know that in most clinical indications, psychiatric or neurologically, we can drive changes that strongly positively empower or improve the quality of life of the individual affected. And still what we do is primitive. But as these strategies are elaborated And as they are applied in more and more careful ways in the medical arena, they will transform neurological and psychiatric medicine. I mean, this is a great practical resource that will be brought to bear in ways that can impact the life of almost everyone on the planet.
So I'd like to go back to something that's implicit in what both Mike and Carl are saying, and that is to a problem that can be studied in crabs. So one of the problems that we study right now, which is directly relevant to the kinds of problems that Mike was talking about, is if you have what appear to be 12 or 20 or 30 healthy adult crabs that all seem to be working really well, then you can ask the question, well, how variable are those nervous systems? How different are two individuals, all of whom appear healthy, one from another? And then you can try and measure all the different components of nerve cell function and synaptic function and say, you know, how many different combinations of these parameters do you have? And can you then predict which of those animals will be more robust to significant environmental change and others less robust to environmental change? So on the one hand, you know, if we had a really strong basic science understanding of animal to animal, person to person variability, at that point that gives you an intellectual handle on the kinds of variability that we see in the human population that makes some people more susceptible to certain kinds of environmental stressors than others.
Okay, so we're almost out of time, but I'm going to ask the question that I sometimes like to ask scientists, which is a bit of a fantasy question, but I have an unlimited grant to give all of you, right? Limitless funds. I'm going to go down the line and ask you, starting with you, Mike, and ending with you, Eve. What would you do in a couple of sentences if you had infinite money?
Well, I would do everything I could to accelerate the translation of the benefits of the science out in the world to help people. And this is going to change the world on the medical side of neurological medicine. And the simple fact is it's taking too long. Because every day that passes, millions of people that could have a better life and that could be safer basically fall off some cliff. So I would do everything I could to accelerate the translation of the science out to help people in need.
Carla.
I have a related answer. I think it would be wonderful to have unlimited funds, because what it would do is let us, and this is for anyone, really continue to work on curiosity-driven research that's not accepted at the present time. And I think we've all experienced, because of our discoveries, which have been out of the box, we've really experienced the agony of rejection. And part of what we do is work hard for years toward acceptance. And I agree with Mike. As I get older, I'm getting worried about cognitive loss and Alzheimer's disease. I mean, the statistics for the aging population are very grim. Over the age of 80, something like over 50% of the population will have severe cognitive loss. And I see that there are connections that could be made between our discoveries and the potential treatments. Our discoveries are out of the box. They do not fit into the current understanding of what's causing Alzheimer's disease or how to treat it. I would work on that problem.
I think you are recognized now. I think that...
Not in Alzheimer's, thank you.
Acceptance,
but, you know,
I think this is part of that process, isn't it?
We'll work together.
It's
We'll
going
work
to
together.
dawn on me sooner or later.
Okay, finally,
I've always worked out of the box. I think for me, the question would be, if there were unlimited money, to fight what I call fighting for the souls of the young people. Because the neuroscience of the future requires an enormously large and extremely well-educated cadre of young people. And what the financial problems of the last 8 or 10 years have done, they've driven a lot of very talented young people out of science. So what I would really, really hope to do would be to develop a new generation of really exciting young creative scientists who are really well trained in quantitative methods, who are not afraid of using and developing new techniques of statistics and new techniques of quantitative thinking so that we can really tackle these and other problems in the future. And I'm just afraid that we're at risking losing a generation, the generation that now is at the graduate school and postdoc era, just because of the practical problems of doing science in the world.
Well, I think that's a very positive message to end on. So all that remains for me to do is to congratulate the three of you, Eve Marder, Carla Schatz and Michael Merzenich.
Thank you.